So this is an interesting area of research right now where we look at how mutations happen in myeloma and where it happens. And we have done whole genome sequences quite deep in a group of myeloma patients and almost analyzed close to 900 plus samples for whole exome sequencing. And we have also included what we call ataxic for epigenomic analysis and RNA sequencing to see which area in the genome are being transcribed, and then integrated all this to understand how mutations might happen. And what we report is that there are certain mutational signatures which are common in myeloma, some of which we have described before, which call AID signature or APOBEC signature or age-related signature. But then we also describe some newer signature which are more DNA repair pathway related signatures. And what is interesting is that different area of the genomes are affected differently by different mutational signature. And what is most prominent here is that the genes which are transcribed, which we have identified through the epigenomic characteristics and RNA sequencing, are more targeted by DNA repair related pathways. And that is great significance because those expressed genes drive the cells and drive the disease. And if they are targeted more by the repair related pathway, we may be able to do some therapeutic applications there. And so the main crux and the message is that transcribed regions are specifically targeted by such processes.